Okay, well, welcome everyone to the Bio 285 review, week 4. This is week 4, number 1, because on Thursday I'll be running another review. I don't know who's running it with me. So this week we are talking about transcriptional regulation, specifically the trip operon and RNA interference. So let's get started. And as always, feel free to stop us if we have any questions, comments, or concerns. So first off, transcription factors. This is the first thing that we talked about. Why do we have them? So transcription factors help to regulate transcription, as you might think, I guess. So transcription factors in, I guess both, but we'll talk about that a little more in depth about eukaryotes. They basically control RNA polymerase binding to promoters in genes. So there's general transcription factors, which are just required for any kind of transcription. So we have in prokaryotes, the sigma factor, which we'll get into. And then in eukaryotes, we have basal transcription factors. And then we have some regulatory transcription factors, which basically positively or negatively regulate transcription, so kind of like activating or inhibiting. So if it positively regulates, we enhance transcription, so we make more transcripts. And if it's negatively regulated, then we're suppressing transcription, so we're making less transcripts. And when I say transcripts, I mean mRNA that's going to go get translated. So that's transcription factors. Specifically sigma 70. This is like my favorite little thing because it looks like a kidney bean. Oh yeah.
Will we look at examples of positive and negative transcription factors, or do we just need to know that? Like I want to.
When we talk about regulation, we're pretty much just going to call them like repressors for the most part. Like we're going to talk about repression and repressors, but you're not going to need to name anything specific unless we give you a specific name.
So we're not going to need it. Like if she gives us like a scenario, we're not going to have to tell her whether it's a. Should we know?
If she were to say something like, "We're making more proteins," you should know that it's a positive regulator.
Okay. Yep. Okay. Got it. Yeah.
Or you know,
I think I was overthinking it.
Yeah, but something like if she gives you a protein and is like, "Here's protein X," and gives it some weird name, and then is like, "It's a positive transcript." A positive regulator of transcription, you should know what that means, but no. Yeah, does that answer your question?
Yes.
Okay, and so sigma 70 is my cute favorite little bean because he looks like a little bean, kidney bean. And we picture it as a kidney bean because it binds at these 2 regions on the DNA. This is called the -10 and -35 spots. That means there are 10 nucleotides and 35 nucleotides behind where transcription starts. And these regions are rich in A's and T's. So you might have heard of the TATA box, which is where, what is it called? Where we bind to originally unwind DNA. And so there's a lot of T's and A's, and that makes sense because if you remember the bonds, but there's only 2 hydrogen bonds between T's and A's versus 3s between C's and G's. So it's easier to unwind there. So sigma 70 is in prokaryotes. Helps RNA polymerase bind. And sigma 70 is sequence specific, which means it binds at certain sequences, and certain protein or certain DNA sequences have better kind of affinity. So if you can imagine, not every DNA sequence has the same letters. So if we're looking here, if you can see my mouse, this is TA, TAAT. Well, maybe this T is something else. Sigma 70 could probably still bind, but it's not going to bind as frequently, just because it's going to bind the most to something that it matches the best, if that makes sense. And that's what a consensus sequence is. And so it binds a lot of genes, but affinity can change. And this is going to become really important when we're talking about like the trip operon and certain other things. So we'll get more into that and some more specific examples. So gene regulation in prokaryotes, operons, which I mentioned today, this week, we're looking at the TRIP operon, which is my favorite topic ever. So an operon is a set of genes that is transcribed under the same promoter. So that's one promoter that transcribes multiple genes. We don't have these in eukaryotes. Polycistronic mRNA is a single mRNA that codes for multiple proteins. So in an operon, we make one big mRNA, and then we translate it and we'll get different proteins. And this picture is great at looking at that. And then again, we control operons by transcription factors, activators, and repressors. This is going to become really important in a lot of the questions we're going to talk about and concepts we're going to go over. So the promoter region is where the RNA polymerase binds, and that is our little purple egg in this case. And then the operator region is where a repressor can bind. So the operator is in front of the promoter, and if you see here, the promoter kind of goes into the operator region, or the RNA polymerase, and it binds, kind of overlaps. So you can imagine if we have a repressor here that's overlapping into the promoter region, our RNA polymerase can't come bind. So physically, it's physically repressing because we can't bind the RNA polymerase in this case. So inducible and repressible. Inducible means we turn it off, or it's usually turned off, but if something comes in, a protein or a molecule or something comes in, then we turn it on. Repressible means it's always on, but it can be turned off if there's a certain molecule or protein. So we're going to talk about the TRIP operon. It's repressible. So the TRIP operon is usually on, but in the presence of tryptophan, it can be turned off. And we'll get into that. So specifically the TRIP operon, tryptophan, if you remember, as an amino acid, it's on your chart. So in low levels, when you have low levels of tryptophan in your cell, we don't have access to tryptophan, which means we need to make tryptophan. Tryptophan is made up of proteins. So the TRIP operon will make all these proteins that make up tryptophan and make the amino acid. So that's when the genes are on, that's when the operon is on. If we have high tryptophan levels, the cell doesn't need tryptophan. So it would be a waste of energy if we were making the proteins required for tryptophan. So in this case, the operon would be turned off. So is the TRIP repressor protein a positive or negative regulator of the operon? Does anyone want to put their answer in the chat or shout out? Is the TRIP repressor protein positive or negative regulator?
Negative.
Yes, it is negative. If we're repressing, then we're stopping it. We're stopping it. We make less transcript. That would be a negative regulator. That's awesome. And then this is a really helpful YouTube video that is linked if you want to watch that, if it's easier to visualize. Okay. I don't know why it did that, but okay. Here's a diagram of the operon. TRIP E, D, C, B, A, those are all your proteins. We're going to get into attenuation, which is Rosella's favorite. And then we have the promoter region, which we talked about, and we have the operator region. And then we have this sequence upstream of the promoter that codes for the tryptophan repressor. And if you look, the repressor, we really simplify this and kind of make it look like Pac-Man. So the repressor does not bind, but if tryptophan is present, it fits in perfectly. We change the shape, so we change the function, and then it can go bind the operator. So that'll make a little more sense as we continue, but that's essentially how that works. If tryptophan can bind the repressor, it makes a complex that then goes and binds the operator, and we repress the operon. So here's some different scenarios, and this is what I would be really comfortable with if I were you during the semester. So scenario one is if we have high levels of tryptophan. What's going to happen to the operon? We're going to have any transcription, and will the protein be made? So what's going to happen with the operon if we have tryptophan? Tryptophan binds the repressor, which in this case, we have tryptophan represented by the little yellow diamond. we activate the repressor and it binds the operative. So this is what we just talked about. So RNA polymerase is blocked. It can't combine. It can't combine, not combine. And then if that can't bind, we don't have any transcription. So we're not making any tryptophan. Does that make sense? Silence, I'm going to just assume means, okay. So scenario 2, attenuation. Now we're going to talk about attenuation. So Rosella, do you want to do the attenuation before I move on with tryptophan?
Sure.
Okay.
Unless you want to do attenuation, it's fine with me.
I don't care. You seemed really excited, so you can do it.
Okay, I have not looked at this slide though. This is the exact slide. Okay, so attenuation occurs when basically you do not have those TERP tRNAs, which causes the ribosome to stay stuck at this region number 1. And so when the ribosome is stuck on this region number 1, it does not allow for the, when it's stuck on the region number 1, it causes the 2,3 hairpin to form, and it doesn't allow the 1,2 and the 3,4 hairpin to form. There's another better image on Laura's,
If you want to, I can go through that. If you want to pull it up.
Yeah, I'm gonna pull it up really quick.
I can talk through this one.
Okay, yeah.
So yeah, so TRP tRNA essentially is, if you remember, tRNAs bring in the amino acids to make the polypeptide chain. So this number 1 region is a TRP codon. So the TRP tRNA would bring in tryptophan. And if we don't have tryptophan in the cell, then there's not going to be enough tRNA. So our RNA polymerase is going to pause here, waiting for our tRNA or tRNA. So then section, you want to switch? Do you want to share?
Yeah, just screen share.
Yeah, you can just kick me out. All right, kicked me out. I think it's funny, every time I'm on a recording this semester, I'm drinking tea. I hope I'm known as the TA that is always drinking tea in review sessions.
Can you guys hear? I mean, sorry, I think my connection is bad, but can you guys see this?
Yeah, I mean, I can see it. That's my favorite picture.
All right, perfect. So that was basically a zoom in of the leader region. So you have the promoter where the RNase binds, the operator where the repressor binds, then you have this leader region and the attenuator, and this comes before the structural genes, because basically what the leader sequence is kind of like a second stop before like the RNA polymerase like commits to transcribing the whole operon. So because basically, so these structural genes, they code for enzymes required for the biosynthesis of tryptophan, as it says here. So basically you don't want to make it if you already have enough tryptophan or enough tryptophan tRNA, which is what causes the attenuation. So let's move on to this. Okay, so here you have the leader RNA. So basically the RNA polymerase starts here. It starts transcribing. This is start site, and this is the start codon for the ribosome. So it starts transcribing, and it transcribes all the way until it gets to the transcription pause. This transcription pause allows the ribosome to start translating this RNA. And so when it gets to this UGG TERP, double TERP codon, it is over region 1, it's over here. And if there is TERP tRNA, the ribosome is going to be able to move on to region 2. But if there isn't TERP tRNA, then it remains on region 1. So when it remains on region 1, when there is not enough TERP tRNA, the RNA polymerase will keep on transcribing. And because the ribosome is over this first region, the 1,2 hairpin and the 3,4 hairpin loop don't form, which would cause attenuation. But instead the 2,3 hairpin forms, which causes the RNA polymerase to keep transcribing the gene. Because if the ribosome is stuck on region 1, that means there is no tRNA, which means that the cell would want to make more tryptophan, so the RNA polymerase needs to transcribe those structural genes. You get attenuation of the sequence and termination of transcription when the ribosome is able to pass through this region 1 and go on to region 2 and so on, because the 1,2 hairpin is allowed to form, and then the 3,4 hairpin also then forms like when the ribosome is here and the RNA polymerase is here. And this 3,4 hairpin kind of physically like makes the RNA polymerase just kind of dissociate from the gene and stop transcribing.
In Laura's terms, it rips it out of the RNA polymerase.
Exactly. I know that is really confusing, but it really helps to just like draw it, like redraw this thing, and just like looking closely at these steps. Are there any questions that I like? Can I go over something that you didn't understand?
There's about 17,000 ways to describe this, and everyone needs it a different way. So if you have questions, please feel free to speak up. And if not, we'll keep going, and we can always come back to this in office hours.
If you like even want me to just like say everything I said again, let me know. That's fine.
Can you repeat it? I think I went to your office hours earlier and you said it once, and it's clicking, but not clicking.
Okay. All right. You know,
do you want me to do it?
Okay. Yeah.
Fine. A different way.
Maybe it'll help like going through each one of these steps maybe, and just like showing.
Yeah, I have a different way, not like a different way, but I think I can hit on it. I don't want to undermine you. I don't want it to seem like I'm undermining you, but sometimes it's just, okay, just want to make sure. Okay, so yeah, it's the 3/4 turning into a fraction for me in the slides. So essentially, like Rosella was saying, transcription and translation occurs at the same time with the trip operon.
Yes.
So, or with any prokaryote. So this region right here that I'm circling, this is the trip region, as I call it because I'm annoying. So the trip region is this region 1 right here. And so this region codes for tryptophan, 2 tryptophans that we need to bring in. So RNA polymerase can go without a problem. And then the ribosome comes in. And when the ribosome comes in, we're bringing in amino acids to make our polypeptide, if you remember, and polypeptide folding and all that comes next. So this is the step 4. So on this trip region, a tRNA brings in, and we actually are going to talk about types of RNA after this, which is weird. We probably should have put that first. But a tRNA is going to bring in the amino acids. So if we don't have tryptophan in the cell, then we don't have tryptophan loaded tRNAs, so we can't bring in tryptophan. So the ribosome stops here, waiting for the tryptophan to come in. 2 and 3, as well as 1 and 2, are complementary. So 2 and 3 can basically pair up because they have complementary sequences. 1 and 2 also can, and 3 and 4 also can, which is where hairpinning is going to come in. So the ribosome will pause here. RNA polymerase transcription pauses here, but that can keep going and we can keep transcribing. So imagine if the ribosome is paused here and we continue transcribing, this too, as soon as—
I don't think I can see your mouse.
Oh.
Cool, it's your mouse.
Okay.
Oh, the 1 region. So imagine that we're on the 1 region. I'll really be explaining it well now, I'll know. So our ribosome is paused there, and then we're transcribing. And so we go past 2 and past the blue. And as soon as we transcribe region 3, it pairs up with 2 and it hairpins, which makes that little fun loop. And we call that a hairpin just because it kind of looks, it actually doesn't really look that much like a hairpin, but we just call it hairpinning. And those can match up. And because it's bigger and it's not as close, to where the RNA polymerase is, it'll eventually just flatten out when the ribosome comes over it and everything is completed as normal. We make tryptophan. And that makes sense because our ribosome paused and we're waiting for tryptophan, and eventually tryptophan will come in, we'll make enough of it, and we'll keep going. Now, if we get to the trip region and there is tryptophan in the cell, then the ribosome, instead of pausing on region 1, is going to go straight to region 2 before RNA polymerase has made region 3. So we went past 1 and 2, the ribosome didn't pause, and so 1 and 2 can hairpin together before 3 is made. If those hairpin, then those are together, obviously. So 2 can't pair with 3. So then 3 and 4 can also hairpin because 3 is free at this point. And that's what the left image looks like. Now, when 3 and 4 hairpin, it's so close to where the RNA polymerase is, it rips it out of the RNA polymerase. The mRNA is literally physically ripped out. And it's called attenuation. So then we just have a short strand of mRNA made, and then we like chew it up, essentially. And we get rid of it because it's not done. And so that happens when we have tryptophan, because we don't want to waste time making all these enzymes when we have tryptophan available. So this is the cell's way, or this operon, this is the way that we kind of can stop transcription from occurring if there's not a repressor bound by some chance, because it didn't happen to bind tryptophan, but we have enough tryptophan, attenuation happens. And this happens a lot in your case scenario when there's like medium levels of tryptophan, because as you can imagine, there's a ton of these operons in the cell, like a ton of them. And so some are going to be going and some are not, just depending on what level of tryptophan we're at. Does that make sense? Is there something you're still confused on?
No, I think I get it, because I think, because if there's enough tryptophan and it stops at the trip,
it's not going to stop for long because we're going to bring in the tryptophan because it's there. So then we keep going. And that's why 1 and 2 can hairpin, because 3 hasn't even been made yet. And the ribosome can move on.
Okay. Oh, because the pause is after the trip, maybe?
Yeah, so the mRNA or the RNA polymerase pauses for a second, or however, it pauses, so we don't make 3 right away. So 1 and 2 are made. So if the ribosome keeps going, then 1 and 2 can hairpin. And RNA polymerase is eventually going to start again. It's going to start again, but not in time. 2 is already hairpinned with 1. So even if we make 3, it can't hairpin because 2 is already doing its thing with 1.
Okay.
It takes a lot to just keep coming back to it 5 times a day. That's my piece of advice for this, but yeah, does that make sense for now? We can come back to it.
Yes, it does.
Do you want to give it some time to digest?
Sure.
Okay.
Okay.
I'm kicking you out.
All right. Sounds good to me.
Okay, so we did this. So scenario 2 is if we have low levels of tryptophan. Now imagine if we have low levels of tryptophan, our repressor is not going to be bound and attenuation isn't occurring because that ribosome pauses. So the 2 and 3 will hairpin if we have low levels of tryptophan, which makes sense because we want to make more tryptophan. So we're not attenuating, we're making the mRNA, we're making the polypeptide, we're making the proteins. And this is the same thing. This is just a different picture. Now, scenario 3 is low to high levels of tryptophan, so medium levels of tryptophan. And then this is essentially what we're just talking about is attenuation. So if we have low levels and then we're making it, eventually we're going to end up with medium levels, if that makes sense, because if we're making it, we're going to get more. So that's when attenuation starts to occur, so that we're not making the same level, we just are making enough. And then eventually, if we have high enough levels, all of them are going to start attenuating until we have enough that they're all repressed. Usually not everything is ever going to be repressed, but in theory, So that's kind of, like I said, that's why attenuation occurs. Does that make sense? I know it's kind of weird because we kind of went through this explanation in the middle of these slides. I did not organize these as well as I thought I did. So yeah, this is what we just went through. That picture that Rosella had up is way better. I would totally recommend that over this because my head is spinning reading this. And then here's your simplified summary. This is a slide to come back to just kind of put in your notes, jot down. Like Rosella said, draw this out, color coding. My page that I drew this out on is my actual favorite page I've ever written of notes ever, because it's all color coded and great. So yeah, high levels, low levels, and then initial low to high. These slides I'm going to send out an email. Oh, there was a question in the chat. Yeah, these don't go on Moodle, but I will send out an email just because I don't have access to upload things onto Moodle. Not that cool. But yeah, we will be sending out these slides along with the recording. Questions? We did questions, so we're going to move on. Thank you for stopping us. Non-coding RNA. I can't find the open reading frame. Could it be a non-coding RNA? It's not that funny unless you're, I mean, I think it's kind of funny, but so non-coding RNAs.
Should I do this? Yes.
Whatever you want. Yeah.
Cool. All right, so non-coding RNAs, as you can kind of decipher, are RNA molecules that don't code for proteins, right? So the RNAs that code for proteins are only the mRNAs. And these non-coding RNAs are super important and they have many different functions and roles. As Laura said, this is the age of RNA.
Age of RNA, baby.
Yeah, exactly. So if you go into like research and stuff, maybe like some sort of cool RNA lab is good, you know.
We know DNA is in a helix. Old news.
Yeah. And so as we have seen in like the ribosome stuff, non-coding RNAs can help catalyze chemical reactions, but they can also regulate gene expression that which is like, we will see an example of this in RNA interference. All right, so now we're gonna look at the different types of non-coding RNAs. So there's ribosomal RNA, which I'm sure you guys know of. It is the catalytic part of the ribosome. So the ribosome is made up of protein and RNA, and its RNA is its catalytic subunit. And a protein or like a molecule that has RNA as its catalytic component is called a ribozyme. So a ribosome is a type of ribozyme. And
they hate us. Biologists hate us when they name things.
And the rRNA catalyzes peptide bond formation. Then we have tRNAs, which basically they transfer, or they have amino acids on them, and they basically come into the ribosome and base pair with the mRNA, and then like the amino acids just like grow in a chain with the rRNA catalyzing it. So this is tRNA, and then the rRNA is somewhere like here in the ribosome. Oh, I did not notice that that was not mine.
You said here, and I'm trying to, this is the tRNA.
Yes. So
here, somewhere here is the catalytic where we're making a peptide bond, I assume.
Yes. And the tRNA has, like, if you look at its structure, you can't really tell in this one, but it has also like a hairpin loop sort of structure, the tRNA. So other non-coding RNAs, there are many different types. We have the small nuclear RNAs, which are involved in splicing. So like their 5 SN RNAs are needed for to make a spliceosome. And then because the catalytic part of the spliceosome is non-coding RNA, it is also classified as a ribozyme. Then we have long non-coding RNAs, and these are involved in X inactivation. It's actually really cool. So these long non-coding RNAs, I think, basically wrap around the X chromosome in order to inactivate it, which is pretty cool. And
Or
then,
also, I'm doing a cancer project right now, and they're
Yeah.
actually looking at long non-coding RNAs as treatments for certain cancers right now, which is also super cool.
that's awesome.
So people go into long non-coding RNA.
And then you have CRISPR RNA. CRISPR also super cool, super important. The RNA acts as a guide strand to target, I guess, invading viral DNA. I have not gotten the chance to work with CRISPR, but my roommate works and like does like experiments with CRISPR, which is so cool. And then you have SRP, which I'm sure you guys are familiar with because it is involved in signal sequencing. It recognizes that N-terminal ER localization signal sequence and brings it to the ER. And then you have RNases, which are everywhere basically, and they're just nucleases that cut RNA. So like, for example, if you need to do like an RNA extraction in a lab, you have to have this like thing called RNase sap, which is just a spray, and you have to spray it literally everywhere, because if you want to extract RNA, which is super like unstable already, but there are also RNases everywhere that will like cut and chew up your RNA. So yeah. Yeah. I do not even know what this is. I don't know what this is.
It's I don't know why it's in a different font, but it's like tsunami, the T is silent, blah, blah. And then at the bottom is RNA interference. The gene is silent. Because the gene is silenced. I totally didn't see that the first time because I only read the first 4. So that's me explaining.
I didn't even see that.
That's us explaining the joke. I hope it was funny and you appreciated it.
Yeah, so RNA interference is basically when you silence the gene. So that's the joke, I guess. RNA interference occurs across eukaryotic cells, and it is a form of post-transcriptional control of mRNA because it suppresses gene expression after the mRNA has been transcribed. And we are going to focus on, so there's like RNA that is involved. A main component of RNA interference is called RISC, and RISC stands for RNA-induced silencing complex. And an important part of RISC is it's a guide RNA strand, basically, which is another form of non-coding RNA. And you have 2 different RNAs that can be like bound to RISC, and you have the microRNA and short interfering RNA, so miRNA and siRNA. And yes, okay. So, all right, I don't love how the chronology of these slides.
The chronology. Do you want me to go on to a different slide?
No, that's fine. That's fine. Okay, so basically we're going to look at 2 different pathways of RNA interference. One is microRNA and one is siRNA. And both of these, they're very similar. And they both involve RISC, which is basically like the spotlight of RNA interference. And basically the general mechanism of RISC is that it binds to RNA, and then this a single-stranded RNA, and this RNA basically is used to as a guide strand to target specific mRNAs. And when it targets these mRNAs, this allows silencing of those of the gene, because if you like degrade or like store the mRNAs before they can get translated, then you're silencing the gene, because like for a gene to like be expressed, like the protein needs to be made. If there's no protein, then even though the gene was transcribed, you don't like, it's not being translated, so the gene is silenced. Yes.
I don't know why there's a questions slide here before we get into the mechanism.
But are there any questions? No. Okay.
If there are, they'll probably be answered shortly.
Okay. All right. So we're going to focus in on microRNA. And it is going to be a guide strand for risk, and it is an endogenously expressed RNA, which means that the microRNA is coded for within the gene, within our genome, and it is transcribed in the nucleus. And it is a really small like segment of RNA, and it like forms the hairpin loop that we saw before in attenuation, which is why it's like it says double-stranded RNA, because the complementary base pairing forms a hairpin loop. Yes, can move on. Okay, so basically, as you can see, the nucleus is in blue. You have your DNA that codes for the pre-miRNA. This diagram is actually wrong. Where it says pre-miRNA at first, yeah, exactly that one. It should say pri-miRNA. So
I crossed it out. You can't see it now.
Okay, perfect. All right, so so basically your mRNA gets transcribed. This is called pri-miRNA, and this gets processed and cut by an enzyme called DROSA. And DROSA basically cuts this miRNA, and when it gets cut by DROSA, then you call it pre-miRNA. So it goes from pri to pre-miRNA. So now you have this pre-mRNA, and this gets exported out of the nucleus with exportin. And then you have this pre-mRNA in the cytosol, which then gets cleaved again by another enzyme called dicer. And then dicer creates the miRNA duplexes. So you went from pri-miRNA to pre-miRNA to the miRNA duplexes. And then what happens is that these duplexes, RISC comes in, gets these duplexes and unwinds the duplexes so that it can get like one single strand of RNA, because you see that the duplexes are double-stranded. So RISC comes in, gets a duplex, and oh, okay, thank you, Isabella. I'm happy. RISC comes in, gets those duplexes, unwinds it so it only gets one strand, right? Because you don't want a double-stranded RNA, because the RNA guide needs to be able to complementary base pair to other mRNAs. So it has its guide strand made from the duplex. Then it uses this guide strand to base pair with mRNAs. And then according to like its complementarity, if it's an exact complement, then Argonaut, which is the catalytic part of RISC, just cuts it and degrades the mRNA. But if, for example, in the other, yeah, that one, that's showing like not complete complementarity between the miRNA and the mRNA. And when that happens, the mRNA gets stored into something called a P-Buddy,
which we touched briefly on. That's we were doing the review session. If anyone remembers the first exam review, I was talking about this pathway for post-translate or post-transcriptional modification, and everyone was like, I don't know your timeout. But we did talk about the P-body, so this is how we get there.
the P-Body.
And that was the best explanation of this anyone has ever given. You're amazing.
Okay. Oh, well, I kind of already said these.
I know. I didn't click through because you were doing such a good job.
Okay, cool. Yeah, I like the pictures better. The picture, yeah. But these are definitely
explanation.
Thank you. So these are definitely like, it's really good to have like these written out when we post it and stuff, so you can like go along. You're good. Yeah. Does anybody have any questions about the miRNA pathway? So mRNA is endogenously expressed. So it starts in the nucleus, right? Gets transcribed, gets cleaved by DROSA, goes out of the nucleus, gets cleaved by dicer. You have these duplexes. RISC unwinds the duplexes, gets one of the single-stranded RNAs. Then it uses it as like a guide to target mRNAs. And then if it's a complete complementarity to the mRNA, then it gets degraded by Argonaut. And then if it's not complete complementarity, then it goes to the p-body.
In
Yes.
humans, it usually goes to the p-body. It's miRNA
Yeah.
usually is not an exact base pair.
Yes, exactly.
siRNA is a different story, but
Oh yes, it says there, remember.
in red, which is the only reason I remember.
All right, now we can go on to the second pathway. So this other pathway is like so similar to the mRNA. It's just like it doesn't do the first thing that miRNA does, which it doesn't get coded for by like our own
Genome.
Thank you. Okay, so basically siRNA, it is also like a small double-stranded RNA, but its source is like exogenous, meaning it comes from outside the cell, and it's usually from a virus. And we use RNA interference and like gene silencing in order to silence these exogenous siRNAs to protect us against like the foreign viral RNA. Yes, and for this one, unlike miRNA, the siRNA pathway is usually like an exact base pairing match between the RISC and the mRNA, which means that usually you'll get cleavage like of the target mRNA instead of like there's no p-body storage. There's only like p-body. I mean, I don't know.
Degradation.
There's no degradation.
It's the nerves. You did such a good job the first time. It's you gotta one up yourself. Okay.
All right. So basically, siRNA pathway, because it's not coded for by our genome, the whole pathway starts in the cytoplasm, right? So you get the siRNA in the cytoplasm, and just like the miRNA pathway, when the pre-miRNA goes in the cytosol, it gets cleaved by dicer, right? So the siRNA gets cleaved by dicer, and you make these duplexes. And then just like the previous pathway, the RISC comes in. You can see that Argonaute catalytic subunit. RISC comes in, unwinds the siRNA duplex, and it uses one of those strands as the guide strand. And then it uses this guide strand to target mRNA, right? And usually when it targets mRNA, there is exact base pairing complementarity, which means that it's going to get silenced and cleaved by the Argonaute, which is this catalytic part of RISC. Does that make sense? So this is just handy dandy comparing, contrasting miRNA and siRNA. So mRNA is endogenous while siRNA is exogenous. And then basically everything is the same except for that the siRNA doesn't have that whole process in the nucleus. It just starts in the cytoplasm, and then it goes like along with what the miRNA pathway is after it gets exported out of the nucleus. Yeah. And then siRNA is always cleavage by Argonaute, while mRNA is usually storage in the p-body. Any questions?
Wish you had been my TA. It made so much sense.
You did very good. You were really good at explaining the attenuation and TERP operon.
Oh, yay.
How
Well, that's all of our slides. How do you later? I have to split. I'm sorry. I love laughing at the memes that we find.
They're great.
They entertain me, and sometimes people are not as entertained, but I'm entertained. Just wait for Thursday. I'm making the slides for Thursday. It's gonna be great.
Perfect.
So many memes. Okay, does anyone have any questions? Anything they want us to go back to before I stop the recording? Okay, well, I'm going to stop the recording here.